okay? So interphase, what happened during interphase? We already briefly talked about in the interphase we have G1, S, and G2. In the S phase, we have DNA replication in the S phase. As you can see that after DNA replication, we go from each chromosome with one chromatid. We become, okay, each chromosome, we have 2 chromatid, okay? And then this a new term I want to tell you is that let me give you the, let's say, green color. Okay, you see that these two, I circle in green, these 2 chromosomes, okay, they look like they are the same length, okay, they're the same length, but we call it one blue, one red, because one chromosome, they are from the father, from your father. The other chromosome, they are from your mother. They are from your mother. Oh, this is bad. Together, we call that homologous chromosome. Homologous. It's like when we talk about evolution, we talk about homology or homologous structure. It is the same word, homologous. We call it homologous chromosomes. They are homologous because they are of the same length and same type. They are the same type. As you can see that in here, we have, I circle it in orange here. This pair of chromosomes, they are of different length. They are shorter, and they are of different type. And they are also, one of them is from father, one of them is from mother. So we call those also a homologous pair, homologous chromosome. So we have 2 homologous chromosomes here. And then at the end of mitosis, you have 2 daughter cells. So we have 2 daughter cells here. So remember, this is mitosis that we were talking about. At the beginning of mitosis, we have 4 chromosomes, okay? At the end of the mitosis, each cell, each daughter cell, we also have 4 chromosomes. In other words, mitosis is that, okay, the 2 daughter cells, they are identical. These 2 daughter cells, they are identical. How do I put it? So these daughter cells, they are identical, okay? And each daughter cell, they are also identical with the mother cell. So they are identical with the mother cell. So that is one of the 2 of the properties of mitosis. One is that, okay, the chromosome number, they are the same. So if at the beginning of mitosis you have 4 chromosomes, at the end of mitosis you also have 4 chromosomes. And then the daughter cell, they are identical with the mother cell, and they are also identical with each other. And then the third point is that mitosis is that one mother cell will become 2 daughter cells. So 3 points, okay? Let me repeat. One mother cell will become 2 daughter cells. Second point, the number of chromosomes is the same. If the mother has 4 chromosomes, the daughter cell will have 4 chromosomes. Number 3, the daughter cell, they are identical to each other, and each daughter cell, they are also identical to the mother cell. So these 3 points, this is like the summary of mitosis. So the first stage of mitosis is called prophase. Prophase, I'm not sure if you can read this, but so these are the properties during prophase. What happened? Okay. During prophase, first thing is that the nuclear envelope disappear. Or you can call it dissolve, nuclear envelope or nuclear membrane. The nuclear membrane disappear. Well, I just put a cross here. Cross meaning it's gone. And then second thing is that you will see a chromosome. Chromosome appear. Why would I call it chromosome appear? Well, this before I would say before S phase, in G1 phase, all the genetic material, the DNA, they are arranged in the form of chromatin. Chromatin, they are thread-like structure. So they are DNA wrapped around. We talked about chromatin before. Previously. When chromatin they wrap around those histone complex, then you form the chromatin. When chromatin condense, they become chromosome. And after S phase, you will have two sister chromatids. Okay. Chromatids. Okay. So that's the this is the S phase. This is the well, I'll say during interphase, the condensation of the chromatin will become chromosome, and then during S phase, chromosome replicate, DNA replicate, then you have two chromatids. Okay. Number three, we talk about the centromeres. No, the centrioles. Centrosome. Okay, centrosome. Centrosome, they start to move apart. So what is centrosome then? I briefly mentioned centrosome in the last lecture, in the last slides. So can you see that there's a structure here, which is the same as here. Basically, you see the two rectangles. They are perpendicular to each other. We call these two centriole. Okay, and then you see that around the centriole, something white around the centriole, so it's like a white circle around the two centriole. So this is the protein gel, some kind of protein gel. The protein gel kind of group them together, and when you have all these together, we call it centrosome. When protein gel and the two centrioles together, then we call it centrosome. So now I hope you understand the relationship. Centriole, they are the two cylinder. They are not rectangle. They're actually cylindrical structure, perpendicular to each other. And then they are these are called centriole, and then they are bathed in the protein gel. And when the protein gel with the two centriole together, then we call the entire assembly centrosome. In here, one of the properties that the centrosome, they start to move apart, they replicate, and they also move apart. Originally, before cell division, you only have one centrosome. During prophase, centrosome replicate and move apart. Okay, so you have these three things occur. Nuclear membrane disappear. You see the chromosome. Originally it is chromatin. Okay, and then centrosome replicate, and then they start to move apart. Metaphase is very simple. It's very easy. The reason is that you can tell metaphase is like a straight line. A straight line in the center of the cell. It's like this. So during metaphase, all chromosomes, they line up in the middle. So it looks like this. They line up in the middle. And you can see that the two centrosomes, right now, they already moved to the opposite end of the cell. What happened is that during metaphase, there will be some kind of spindle fiber. There's some kind of a spindle. They will project some kind of microtubule, those spindle fiber, and then they'll attach to the centromere. Do you still remember centromere? Centromere is at the center, in the middle of the chromosome. Those spindle fiber will attach to the centromere, and then there will be like a tug war. The two centrosomes will start to contract the spindle fiber, those microtubule, the spindle fiber, so that they will pull the chromosome from opposite end until they line up in the center, the equatorial plate. The center, they call it, some textbook they call it a metaphase plate. Some textbook call it equatorial plate. It's like the equator. So that's why all the chromosomes, they will line up in the middle. Okay, anaphase. Anaphase basically is that the centrosome continue to shorten the length of the spindle fiber, the microtubule, so that it pull apart the two chromatids. The two chromatids separate. So anaphase is basically the separation of the separation of two chromatids. If you look under microscope, it looks like two bananas. Two bananas, they are two, it's like, two fingers, two hands, they are facing each other. Two hands, they are facing each other. So we call this the anaphase. Telophase. Okay, telophase is basically, you see the reforming of the nucleus membrane. The nuclear membrane reform. Oops. Nuclear membrane reform, and then for animal cell, okay, animal cell, for animal cell, you have something called cleavage furrow here. Cleavage furrow. show up. Cleavage furrow is because there is a contraction ring here. There's a contraction ring here. It's basically the cytoskeleton. They contract and then pinch the cell so that it forms something called cleavage furrow in the animal cell. But what about plant cell? What happened to the plant cell? The plant cell, it has something called a cell plate is formed. Cell plate is basically, you can see it's like a straight line in between the two future cells. And this cell plate will become cell wall. You will not see cleavage furrow in the plant cell because they have cell wall. It is rigid, but you will see the cell plate. It is the future cell wall that separates the two cells. So we finished